what a typical bulk HiC data set might look like. White color is zero interaction, red is high interaction. So here you have the heterochromatin region, that's why you have this band, but otherwise you are seeing that, you know, on diagonals, close to diagonals, you have a lot of interactions. And as you go away from the diagonals, you see fewer interactions. And then if you look at a high depth HiC data sets coming from the same system, for a single cell, the data might look like this. Much more sparser, but the general characteristics, especially these block diagonals, are captured. Obviously, if you decrease sequencing, then you might even end up something like this. So this would be data for one chromosome of a single cell. So right now, with this technology, we can only reliably capture intrachromosomal interactions. So we don't have the depth to capture interchromosomal interactions. So that means that we can, for a given chromosome, we can stack up these matrices of contact counts, contact matrices, and then obtain these three-dimensional tensor structure. So this is, you know, like extending the typical cells by genes to a slightly more complicated data structure. Okay? And with this type of data, main analysis objectives are the usual. So given, you know, if the data is coming from a complex tissue, we want to identify cell types, cell subtypes. We want to ask whether just based on 3D chromatin structure, can we learn differences in cell types and subtypes. But beyond that, once you have cell type specific contact matrices, then you want to identify loops, which are interactions between any two genomic locus. You want to identify topologically associating domains, big regions of the genome that have a lot of interaction, and then AB compartments of the genome, which are megabase regions that delineate active and inactive chromatin. Okay? And then, of course, there is the multimodal data integration for this data type. And data integration in single-cell HiC is obviously huge, we have attacked this problem integrating HiC data with other data types using tensor decompositions. So the first half of my talk is going to talk about this method that we developed that is based on leveraging the tensor structure of the HiC data. Okay, given the tensor structure, we utilize what's called a block term tensor decomposition, which essentially models the tensor structure as low-rank modules. So let's assume this is a tensor for one chromosome. So again, these slices are cells, and cells, contact matrices of the cells, and this is one chromosome. If you are looking at chromosome 1, and if you have two low-rank modules, then it's the sum of these two objects. These are the low-rank structures, and in this low-rank structure, the first component is cell loadings, which is number of cells by one. It's a vector, a non-negative vector. And then the second component is loci loadings, which is number of loci, the artificial bins that we create on the genome, by k, some low dimensional, some low dimension k. And in this context, these loci loadings actually capture the, ideally they should actually capture the cell type specific contact matrices, cell type specific interactions. And here, so the cells are going to be common across, cell loadings are going to be common across all chromosomes. So that's information sharing across chromosomes, because we have the observations coming from a single cell and then contributing to multiple chromosomes. And then loci loadings are going to be obviously chromosome specific. So if you know a little bit about tensor decomposition, you might ask, you know, why not some simpler decompositions, CP So there's a known popular tensor decomposition called CP decomposition, which essentially writes down the tensor as a outer multiplication of unit vectors. So in this case, you would still have your C, but A and B, instead of being low rank matrices, they would be vectors themselves. We actually did start with this, but it turns out that the contact counts are not rank 1. You need more rank to represent those. So if you go with something like this, then you would have many more modules to actually capture the contact counts for cell types. And then it becomes harder to attribute individual modules to individual cell types or subcell types. And then another way to think about this decomposition is that, again, if you know about Tucker decomposition, which writes the tensors as a lower dimensional core tensor multiplied with 3 matrices, then block term decomposition is a special parameterization of Tucker decomposition where the core tensor is block diagonal. Okay. So once we have, the thing about this framework is that once we have the decomposition for one chromosome, then we can actually have the decomposition for all the other chromosomes. Again, the loci loadings are going to be chromosome specific, and the cell loadings are going to be shared across chromosomes. So this naturally allows a framework where you can integrate multiple modalities. So that brings us to methylation, DNA methylations measured at the single cell level, usually coupled with scHiC data. So especially the high single cell scHiC data coming from the Brain Initiative is multimodal. So for each cell, you both have 3D structure measured by a scHiC, but also DNA methylation at the single cell level. Okay, and the methylation, as you probably all know, is a very important epigenetic mark that's especially involved in gene expression repression. And then methylation is usually summarized as a matrix. If the resolution is there, it could be rows are cells, columns are individual CPGs. For some tissues, non-CPG methylation is important. So CPG is C followed by a G, but in brain tissues, non-CPG methylation is actually quite important. So you might have 2 matrices, and if the resolution is not there, usually methylation is also summarized at a certain bin level. Okay? So then we can naturally, and then, a matrix is just a rank 2, dimension 2 tensor. So it easily, this framework easily encompasses integration with the methylation. So then we add the methylation data as as for each, as R modules. Again, CR is common across it's common across data types and also data types, these 2 data types, and also chromosomes. And then we have a loci loading for methylation that delineates the methylation profile along the genome, or whatever your experimental unit is here. And then finally, we can write down the whole model as the composition of these 2 tensors. So I already talked about the importance of C cell loadings being common across chromosomes and also data types. So that's how we succeed in integrating. And also we impose non-negativity constraint on the C, which then also helps with the interpretation for a given loading, cell loading. If you see that, it has high values for a subset of the cells and really low values for the rest of the cells, then you can ask, well, could this be corresponding to a cell type or a subcell type? So that really helps with the interpretation. So that's the first component, important component of this model. And then another important component is scHiC and methylation. They are measured at different scales. They have different, sizes in terms of the experimental units that they are contributing. So it's important to do some sort of scale normalization. And instead of doing something arbitrary, we actually use a proportional variance assumption where the error variance is proportional to the sizes of the tensors, scHiC, single-cell scHiC data tensor, and the methylation tensor. Okay. Then we can write this nicely as a minimization problem. It's a non-convex problem, but we can use, we were able to develop an alternating least squares algorithm where, given cell loadings, we can estimate chromosome and data type specific loci loadings, and then given loci loadings, we can estimate cell loadings. So we can do these updates in an optimal way. Okay, so before I show you some results, again, there are probably a gazillion different ways one can attack this problem, but one nice thing about this formulation is that all of these parameters that fall out of this formulation correspond to very natural downstream analysis that you would want to carry out with scHiC data. So we already talked about cell loadings and how you can use cell loadings to learn something about the cell types. Loci loadings, you can think of it as an object that's already denoised that you can use to infer topologically associating domains. And again, sign of the first column of the loci loading, which is already denoised now, can be used to learn AB compartments. And then loci loadings, the multiplication of these vectors give you denoised version of cell type specific or module specific contact counts, which if you know whether certain modules are corresponding to certain cell types, then you immediately get the contact counts. And then once you estimate these things, we can do correlative analysis in terms of what kind of methylation profiles correspond to what kind of long-range interactions. So again, we all work in single cell domain, right? So there are very many different ways of doing these type of analysis. But the nice thing about this formulation is that it automatically gives you the objects that you would need for different type of downstream analysis of scHiC data. Okay, so I won't showcase examples of these. The paper is published, and then we have detailed examples and discussions of how this framework achieves all of these tasks. I'll just show you a showcase how this performs compared to some other methods and how it is leveraging multimodal data. So this is an old data set. It's kind of a proof of principle data sets where they mixed up different cell lines together, and then we applied a number of different methods. Some of these actually were also developed in my group, and most of these methods work by estimating some sort of lower dimensional structure for the cells, and then you typically visualize this with UMAP. And as we can see, most methods are doing a pretty good job in terms of getting the major cell lines here. Everyone is struggling with IMR 90 because there are so few IMR 90 cells in this setting. And then most notably, scHiC topics, 3DVI, and muscle are performing pretty well in terms of separating these cell types. So this is one showcase. When we apply these methods to many other single-cell scHiC data sets and evaluate using different metrics, and then look at the rank of these methods, we see that many methods actually perform pretty similarly, and then maybe muscle ekes out with a slightly better rank. Okay, so now I actually want to show you where we truly have a multimodal setting, where we have 3D genome and DNA methylation from human prefrontal cortex. This is obviously a much more realistic and difficult data set because we are looking at a real tissue. And then I have here, so there aren't many methods that can analyze this type of multimodal data where one of the modalities is scHiC. There's a method called Higashi, but it's notoriously slow to run, so I'm going to only show you muscle results. So we first applied muscle to only scHiC component, CpG methylation component, and then non-CpG methylation component. And then in this data set, there are neurons and then subneurons, excitatory and inhibitory neurons, and non-neuronal cells. So first, we see that if you just use the scHiC component, actually it's the subtypes of neurons are hard to separate. If you look at, let me see, hard to separate just before the scHiC component, but non-CpG methylation actually does a pretty decent job of separating these. In contrast, if you look at the scHiC component, oligodendrocytes, the two big clusters of oligodendrocytes are separated nicely. However, both the CpG and non-CpG methylation have a hard time separating them. If you put everything together, if you analyze these data sets jointly, actually you are borrowing information from both modalities and you are able to separate all of them. So we compared this with a baseline approach where you can either flatten out the tensor and then treat it as a matrix and apply dimension reduction and then combine it with the methylation component, or you can apply one of the existing methods on the scHiC component so that you are learning the lower dimensional structure in a way that respects the data structure and then combine the resulting loadings. And in all of those cases, actually, the baseline approach doesn't do better than muscle. And then if we look at data sets, so this is another more recent data set which has this multimodality, using all modalities with muscle does better than the other baseline approach, or using individual modalities. So I'm gonna summarize this. So the real contribution is just recognizing the tensor structure and then utilizing data decomposition that respects the tensor structure. And we have optimality properties for the algorithm that we use to estimate these parameters. But again, as I emphasized, we are most excited about the interpretation because there's a unification of estimation target and how you are parameterizing your model. So that's all I want to say about muscle. We can discuss it. We'll have time for questions if you have questions on that. Now I want to move on to, so, with single cell scHiC, we can learn 3D structures at the single cell level, combine it with different data types. But what else can we do? So this is a question that we have been thinking about for a long time. And then 2 years ago, there was a science paper that was published that leveraged these perturbation CRISPR experiments. So they essentially looked at, in K562 cells, this MYC gene, which is a key oncogene governing cell proliferation. And the activities of this gene is regulated by 4 enhancers that's immediately downstream of the gene, and then another 3 enhancers that are almost 2 megabases away. So this is known from the literature that all of these enhancers play a role. So they wanted, this paper essentially wanted to know what are the interactions between these enhancers. So they designed guide RNAs that either target these enhancers individually or they target them in pairs. So, and then what they found out is that once they target either individually or in pairs, if the enhancers are in the same region, their effect is additive. So what I mean by that is that, so in these experiments, they are measuring gene expression as an output. So when they target when they delete, knock out E1, this is the reduction in expression. When they knock out E4, this is the reduction in expression. So the expression level drops here. When they knock out both, that's what you observe as expression. And then this purple line is what you would expect, the region you would expect under an additive model. And same for two enhancers that are in the distal region. If you knock out E5, you get some reduction. E7, you get some reduction. But if you knock out both, again, the effect is additive. So that's all good. But then what they found out is that if they knock out one from this region and one from this region, the reduction in the expression can no longer be explained by an additive effect. So there's an interaction effect between the enhancers from these regions. So if you knock out E3, that's what you get. That's the reduction expression. E7, that's the reduction. If you knock out both, now you observe a reduction that's much larger than you would have expected under an additive model. Okay. So this made us this made us think that, okay, if you think about GWAS and other type of molecular QTL studies, I mean, our sample sizes are increasing, right? But we are not talking much about epistatic SNP interactions. However, if you think about this case, you know, if you have a SNP, you know, disrupting E3 and a SNP here disrupting E7, so you would expect an interacting SNP effect, epistatic effect between those two SNPs. Okay? So, you know, even though our sample sizes are increasing, we have more and more SNPs, rare SNPs. So looking for these type of interactions is actually still very hard. So then we thought if there was a way to, you know, to generate candidate multi-way interactions, okay? Candidate interaction regions, so that we can, when we look at GWAS or molecular QTL, we can go look for these interactions in a very targeted way. So then this kind of asks or begs the question, can we identify multi-way interactions? Okay. So when we think about scHiC data, that's all pairwise, right? I have the contact matrix and the entries tell me, you know, how much any two genomic loci are interacting. So with multi-way interactions, we are almost looking for, you know, at least 3 genomic loci that are interacting. And then ideally, one of them is the promoter of a gene, and the other 2 are enhancers. Okay? So there are indeed, in the last few years, special technologies to profile these type of multi-way interactions. I have a list of them here, and they all have proof of principle in different cell lines. But I would still consider them as boutique technologies because, you know, there's one paper and then maybe 2 data sets on that type of technology. So then we ask, is there a way to learn multi-way interactions from scHiC data or single-cell scHiC data? Okay? So initially, you might think of this as, okay, if I'm asking, are these 3 loci interacting? If I see all 3 pairwise interactions, right, then they must be interacting. So this is what I would call a multi-way interaction. We have all the pairwise interactions, so then the whole structure must be interacting together. Okay? But so, you know, in the network terminology, this, you might think of this as a clique where the graph or subgraph is fully connected. However, the sort of bad news is that scHiC works with proximity ligation, and all the scHiC data sets thus far are based on short read sequencing. So if you have a cell and then these 3 are interacting, if this is the fragment you are capturing, ligation happens at random, so you might end up having these 2, purple and pink ligated, and then here pink and green ligated. So that means that you'll have this interaction, but not this one. So you are unlikely to observe full cliques. However, if you really think about this, if you are looking for this type of interaction, a 3-way interaction, you know, as long as we have 2 of the interactions, we can infer that these 3 loci must be interacting. So that was the insight we had, and then we said, let's actually look at the actual single cell HiC data and ask whether we are seeing cliques. And then we did this for a number of different single cell HiC data sets at low resolution and high resolution, and then you see quite a lot of cliques, lots of three-way cliques, and then fewer four-way, five-way, and six-way cliques. And you rarely see more than six-way cliques. But that's still pretty promising because, you know, if we were to figure out which of these cliques are not happening by chance, then we would have our candidate multi-way interactions that we can utilize to interrogate GWAS or molecular QTL data sets. Okay, so if we can enumerate these, which we can, then the question becomes, given a candidate clique, can we identify whether the interaction, all the pairwise interactions we are seeing in that clique, is that you know, much higher than expected by chance? So it just now becomes calculating some sort of statistical criteria for quantifying that. And a natural way to do that is to think about a null model for pairwise interactions. And so the mother of all models for count data or sequencing data, you can think of, you know, the whole matrix coming from a giant multinomial. So that's the natural starting model. And then one thing to pay attention here is that HiC has what's called a distance bias. So the number of counts you observe any two loci is going to be a function of their actual genomic distance. So this is very well documented. So to handle that, usually it's very natural to summarize the data at the band level. So my notation here kind of supports that. So that's just conceptually, you know, how we are putting down the terminology. But if you wanted to fit this model where these pi's depend on the genomic distance, and usually this is nonlinear dependence, you would want to use some sort of nonlinear function, maybe a spline. Just fitting that is computationally very intensive. If you have, you know, your single cell HiC count matrix is at 10 base pair resolution. Now we are, you know, for chromosome 1, this is going to be in the millions. But what we can do is a simple trick. We can do multinomial aggregation at the band level, okay, and then Poisson approximation to multinomial. If we do that, then the counts that you observe at the bands just become Poisson with a mean parameter that depends on the sequencing depth of the cell and also the band level interaction probability. So, and then that's a function of the distance between any two loci in that band. Okay? So we use a spline model to fit that, and then this is now much more easier to computationally fit, and then we can estimate these band level probabilities Pj. However, to go to interactions between any two loci, we still need locus level probability, locus pair level probabilities, and for that we assume that the locus level probability, so this is the probability of interaction between locus I and J, it depends on the band level Pj, which we estimate from this model, and a Dirichlet prior. And this prior can depend on the underlying mappability and GC of those locus pairs. Actually, if you have single cell HiC data with multiple cell types, there are easier ways to estimate that. Okay. So with that, we can actually estimate a null probability of interaction. And then given that, we can easily calculate for a given edge in a multi-way interaction, the expectation and the variance under the null. All of these are analytical. And then by aggregating across cells, k equals 1 to k, we can calculate the summary statistics or a z-score for this edge. Okay. So, you know, what is the evidence for this edge deviating from the null is quantified by this z-score. And this behaves very nicely. It has normal 0, 1 distribution, and then these edge p-values corresponding edge p-values have uniform distribution. So this is again now just edge level statistics. If you wanted to call loops, for example, this is a quantity that we can use. However, of course, we want to raise this to a clique level score. Okay? So we could aggregate all these pairwise, do different type of statistics, which we did. But it turns out that if you are thinking about a multi-way clique, we don't need all the pairwise interactions to be extremely strong. So we just need certain number of them. So for a three-way interaction, just having 2 strong pairwise interactions is good enough. Okay? So then, as summary statistics for the clique, we use at least R number of them should be strong. So that's the Rth highest z-score or Rth smallest p-value. And it turns out that theoretically, if you have p-values coming from a uniform distribution, which they should be under the null, and then if you look at the Rth one, that has a beta distribution. So again, we tried many different statistics to combine things at the clique level. This worked out really well. Here I have results for a particular cell type. This is again a prefrontal cortex data set. So these are, if you look at the QQ plot, the y-axis is the observed p-values, so that's coming from the beta distribution at the clique level, and then these are the expected. Okay? So then these p-values, each of these points correspond to a type of multi-way interaction. Okay. So then, of course, we want to see how this method works. Ideally, you want to apply it to many different data sets, and then you want to think of an orthogonal data set that can help you validate the results. And ideally, that orthogonal data set should be something that measures the multi-way interactions directly, like Sprite, GEM, all these boutique technologies that I talked about. But for the data sets that we had, that we could apply this to, we didn't have that type of multi-way interaction data set for all of them. So we had to come up with different ways of doing it. I'll show you a few examples. So this is, so all the applications that I'm gonna show are prefrontal cortex applications. Actually, not all of them. So, okay, I'm starting with GM cell lines. For GM, we also had Sprite. So we call multi-way interactions. We have our p-values. We can apply FDR control. So then we can say, okay, these are statistically significant interactions. These are not. And then you would expect that the Sprite score for those should be higher than the ones that are not significant. And that happens to be the case. And then if you sequence scHiC data with long reads, then actually you can get, from the ligation products, you can get multi-way interactions. And this is a newer technology. There's only 2 data sets that have that property, but with that we were able to show that indeed the multi-way interactions we infer with MinC is have higher scores when evaluated on scHiC data set that are coming from long reads. And then another data set that is useful for validation is DNA-seq fish. This is again, a lot of this is coming from the 4D nucleome. It's a little bit like spatial transcriptomics, but for chromatin and 3D structure. Essentially, you can profile the location of individual loci within a cell, and then you can calculate the spatial distance. So that means that, high MinC score should correspond to low spatial distance, and that happens to be the case, although it's the signal is not very strong. So in order to make sure that this wasn't an artifact, we did a lot of permutations, and if you permute, you never end up seeing this kind of decreasing trend, which indicates that what we are seeing here, although it seems like low signal, is actually real and supporting the multi-way interactions. Okay, so then I'm going to tie it back to why we want to capture these multi-way interactions. The idea is to be able to go in and profile molecular QTLs or GWAS. So this is ongoing work in collaboration with Jingwen Yan. We took an Alzheimer's study where you have controls and Alzheimer's and expression, gene expression from their cortex. And then we took the multiple interactions that we inferred, and then we ask if we consider interactions of this type where we have a gene promoter and then two other enhancers or intronic elements. Then we try to explain the variation in the expression of this gene as just an additive model of the SNPs that are in these locus, in these loci, or with an additional interaction term. Okay, so we tested for this effect, and that's the QQ plot. The red ones are all the interactions we have tested, and the gray is if you permute the data and then ask, what the null looks like. So we see that all the interactions we have tested are actually deviating, and furthermore, some of them actually make the multiple testing cutoff. So here is one of the interactions for actually DKK3 promoter. There are two enhancers, and these are enhancers in its intronic regions because we also have single cell scHiC data. We can look at these loci and see whether there's a regulatory signal there. And then we see that if you don't have any minor allele, this is what the adjusted expression looks like. If you have one minor allele, maybe there is little drop in expression. If you have minor allele from the other SNP, you don't see much change. But if you have both minor alleles, now you actually see a significant difference in expression. So this is the kind of thing that we want to be able to do, and multiple interactions, capturing them, utilizing scHiC data, at least gives us a starting point to do that. Okay, so this is my almost the last slide. So I talked about utilizing scHiC data beyond, learning pairwise interactions or doing the usual stuff that we do. So I think their real value is to be able to utilize them for probing epistatic effects. And also, I would like to bring to your attention, so we have a lot of molecular phenotypes for cells, accessibility, transcriptome, 3D organization, methylation, so on and so forth. But we are also starting to see phenotypes at the cell level. For a given cell, you might have its electrophysiological profile, you might have its morphology. Then, of course, the question becomes, can we utilize these molecular phenotypes to learn something about these phenotypes that are at the cell level? So that's, I would say that's the one of the newer challenges where we have at the cell level high dimensional phenotypes, but high dimensional variables that we can potentially utilize to explore these phenotypes. Okay. So with that, I'm gonna thank Kwang Moon Park, who is the first author on both of these works. He's phenomenal. He's on the job market for a postdoc, so I don't know if anyone is hiring. And then scHiC is the second student in my group who works on these problems. And for the multiple interaction, we doubted ourselves for a long time whether or not we could really do this. So we had a lot of external consultants that helped us work through this. Okay, and with that, thank you for your attention. I'm happy to take any questions you might have.
Very nice. Given the structural sparsity, the high sparsity of single cell scHiC data, have you ever thought about adding a third dimension to muscle in the dimension reduction where that would be the local neighborhood? For instance, think of a Z-stack of cells and they're ordered by their local neighborhood, which would allow you to impute along axes of signal that are conserved in the local neighborhood but maybe dropped out in that particular cell. Because you have a very nice two-dimensional alternatingly squares problem.
I have ringing in my ear. and it's just coming down.
I'm sorry.
Could you repeat the question? Yeah.
I'm asking about adding a third dimension to your two-dimensional alternating least squares problem in muscle, where you have not only the local connectivity within that cell, but now also a conserved pattern between cells. So if you have a Z-stack of the cells in the local neighborhood, now your non-negativity constraints can allow you to impute across conserved information in the local neighborhood rather than just within the cell.
We haven't thought about that.
Okay.
I think that sounds like a good idea. Yeah. So borrowing information to impute across cells. Absolutely.
So my question is also in regards to muscle. And you showed really nicely how integrating across these two information types allows you to get additive resolution. But you can also imagine a situation where imputing across the two information types leads to destructive resolution. Have you seen any evidence of that? And like, what's kind of the variability that you see in terms of clustering in one type of information to the other?
So that's a good question. So we thought a lot about that. There is really no way to quantify that because often you don't have a ground truth. So, this, I think one should purely approach from a data analysis point of view, do individual modalities, and then do both modalities, and then really compare the results and dig in. So there is really, we can always put unrealistic assumptions and then come up with something that with the two data sets that we worked, and then one of them is a really decent data set from the GAIN initiative, there was no way of doing that. But that's a good question. Yeah.
Thanks.
Hi. It sounds like the computational framework you've come up with here is very general. Have you played around with any other data types, such as scRNA-seq or ATAC-seq?
We haven't. That would be interesting to do. So yeah, we are really going strong in scHiC, and we moved on to really trying to utilize scHiC to learn more biology. That's also probably an artifact of that. Yeah. But so if you have RNA-seq, then it's like you have non-negative matrix factorization. It kind of reduces to that if you don't have the cancer structure. Yeah.
Thank you.
This will be our last question from a member of our online audience. What advice would you give a collaborator to power an experiment for finding these higher order clicks in terms of sample size, seq depth, etc.?
Sample size. So I think so that's good. I can, for the prefrontal cortex, for some neuronal data types, we only have maybe 50 cells or so to learn these interactions. So sequencing that, I don't know how much control people really have on those and how much, they try to maximize that. But I would say probably getting more cells, even if they are more shallow, is better because you can do imputation. In terms of the actual statistics, what matters is you want to have enough sample size so that you get a good z-score for the edges. Then you can aggregate to click level. And for prefrontal cortex, if you have 50 cells or so, that works really well. So we were able to, validate multiple interactions that are learned from those type of sample sizes by orthogonal data. Mm-hmm. All right. Yeah. Thank you.
We've got a 15-minute break. Sorry, 5-minute break. 10-minute break. Sooner or later, I'll get an approximation of it. So at 9 o'clock, right, we'll resume. And as it happens, we'll resume by another talk on non-negative factorization. So see you then.